In this video, I want to talk about post-transcriptional modifications. So these are ways in which the RNA that's been transcribed is modified in some way. So in prokaryotes and eukaryotes, they both have their RNA and tRNAs processed in a certain way, and actually pretty much the same way. Both the rRNA and the tRNA are cut and trimmed. Essentially, the initial transcripts are a little bit too big, and so in certain regions they're cut around and trimmed to be a little bit shorter. No details really are associated with that. At least I'm not going to talk about them. The tRNAs have a further modification. There is an addition of a CCA at the 3' end. So you add 2 cytosines and an adenine at the 3' end. So if this is a tRNA, we're going to be adding a C, and an A here at the 3' end of a tRNA. And what's the reason for that? This CCA is important because it is the attachment site of amino acids that are going to be incorporated in translation. So amino acids will actually attach to this CCA end right here. So there'll be an amino acid attached there, and the tRNA will bring it to the ribosome for translation, but we'll talk more about that later. Now, eukaryotes have their mRNA modified. Prokaryotes don't. So I'm going to talk about the eukaryotic mRNA modifications. The first one is the 5' cap, and the 5' cap is a 7-methylguanosine cap. And it's attached via a 5' to 5' triphosphate linkage. What does any of that mean? Well, the 5' indicates that if we have a particular strand of mRNA, the 5' end is going to be capped in some way. That cap is a 7-methylguanosine, and it's connected via 5' to 5' triphosphate linkage. So what does that look like? Well, if we imagine this to be the mRNA, we have the ribose and a base. This is the sugar phosphate backbone here. Sugar, phosphate, sugar, phosphate, sugar, blah, 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 blah. And these are the bases over here. So if this is the 5' end, right, these phosphates are attached to the 5' end. So if this is the 5' end of a particular strand, what are we going to have? Well, this here, this portion here, let me actually outline it, is the 7-methylguanosine cap right here. Okay, so why is it called a 7-methylguanosine? Well, this portion is a ribose with a guanine attached to it, so that's the guanosine portion. Why is it called 7-methyl? Well, because if you go through and number the purine, this nitrogen is atom number 7. So that 7th member of the rings has a methyl attached to it, so it's a 7-methylguanosine. And it's attached to the mRNA strand via a 5' to 5' triphosphate linkage. So this ribose's phosphate groups, or this nucleotides, or this guanosine, is attached to this ribose, right, with a 5' phosphate attached to this 5' phosphate. So, and there's 3 of these phosphates here. So that is a 5' to 5' linkage, and there are 3 phosphates. What is the purpose of this 5' cap? Well, it does a few things. First is that it aids in transport from the nucleus to the cytoplasm, or the cytosol. That's important, right? Because in eukaryotes, transcription occurs in the nucleus. Another thing that it does is that it protects the 5' end from degradation by nucleases. If you recall, nucleases break down nucleic acids. So any exonucleases that want to act on this end here won't be able to because of this 5' cap. Another thing that it does is that it promotes the binding of the mRNA transcript to the ribosome for translation. So the mRNA needs to be translated, right? That's the purpose of mRNA. So it needs to be translated on a ribosome. Well, it needs to somehow bind the ribosome. So this 5' cap helps it do that. Another modification is a 3', let me actually write it in blue, a 3' poly-A tail. So lots of adenines are attached to the 3' end. So it's a poly-A tail, and it's attached by polyadenylate polymerase. That makes sense. It's the thing that adds a bunch of A's on it. So if this is a 5' end of an mRNA, and this is the 3' end, we add a bunch of adenine nucleotides, or the letter N here to represent a certain number of nucleotides being added. What's the purpose of this 3' poly-A tail? Essentially, it protects the 3' end from degradation. Oops, that's messy. From degradation. And usually there are about like 100 to 300 A's that are added to that 3' end. The third modification to mRNA in eukaryotes is splicing. The mRNA transcripts are spliced. This is carried out with this thing called the spliceosome. Which contains these things called SNRPs. S-N-R-N-P. SNRPs. Okay, that's a funny name. I actually enjoy saying it. SNRPs, that stands for small nuclear RNAs and proteins. So when splicing occurs, there's this thing called a lariat structure that's formed, and I'll show you exactly what that looks like in just a second. But what's happening in splicing is that there are these things called introns and there are these things called exons. So introns are intruders. So what happens to them is they are cut out. Don't let that confuse you. Exons are exceptional, right? They're the exceptions. Oops. I could just write exceptions, or exceptional, however you want to remember it. But these exons, they stay in. So the introns are spliced out, the exons stay in. How does that actually happen? Well, if we have, for instance, a pre-mRNA or an immature mRNA with exon number 1 here, and then some intron, and then exon number 2. On these introns, there are these certain branch points. Now, what these branch points are involved in is the formation of the lariat structure that I'm about to show you. So, this branch point, what happens is it's 2' OH, right, on the ribose ring of a certain nucleotide over here, is going to go over and nucleophilically attack here at the it's going to attack with the nucleotide over here on the intron that's attached to exon number 1. And when it does that, it bonds to itself. And that's what this is. It kind of loops around and forms this little... Actually, probably shouldn't have drawn that arrow in that color so as to not be confusing. Let me draw it like this in blue. So it binds there, and it kind of loops around like this and binds itself at that branch point. So that branch point here is right there. Then exon number 1 now has a free 3' OH group. Now what happens to that is that 3' OH group goes ahead and it nucleophilically attacks here at the exon to cause this lariat. So let me actually label that. This lariat structure of the intron to break off, and then the exon 1 gets to connect to exon 2. So what happens is the exon number 1 and exon number 2 become connected, and then this lariat structure dissociates away. So now we went from a pre-mRNA or immature RNA to a spliced and mature mRNA. So that's how splicing works. Now, maybe you thought about this earlier, but why don't prokaryotes have post-transcriptional modifications of mRNA? Well, to answer that, we have to answer these questions here. So, do prokaryotes have a nucleus? No. Do eukaryotes have a nucleus? Yes. Now, where does transcription occur in each of these types of cells, or each of these classes? In prokaryotes, transcription occurs in the cytosol. And the reason why is because prokaryotes do not have any membrane-bound organelles. They don't have a nucleus. In eukaryotes, transcription does occur in the nucleus. Now, where does translation occur? Translation occurs in the cytosol. In both of them, prokaryotes and eukaryotes. In prokaryotes, by default, really, because they have no place other than the cytosol for things to happen. In eukaryotes, translation occurs in the cytosol, transcription occurs in the nucleus. Now, why is this important? Because in prokaryotes and eukaryotes, we have to think about, what about the timing of transcription and translation? Well, in prokaryotes, transcription occurs in the cytosol and translation occurs in the cytosol. So as soon as an mRNA is transcribed, the ribosomes in the cytosol can hop right on and begin translating away. So the timing in prokaryotes is at the same time. So essentially what's going on, it's actually a little bit messy, sorry about that, at the same time. So the mRNA doesn't have a chance to get modified. It doesn't have time to get modified. Because as soon as it's transcribed, it begins to be translated because the ribosomes are in the cytosol. In eukaryotes, however, it's a little bit different. The transcription occurs in the nucleus, so the mRNA is made in the nucleus, and it has to leave the nucleus to go to the cytosol and then be translated. So here, they're at different times. Because the mRNA has to leave the nucleus through the nuclear pores and then enter into the cytosol. So then translation can occur. So because the mRNA is sort of separated from the cytosol by the nuclear membrane, here there is time for modifications, essentially. So that subtle difference of, the presence or lack thereof of a nucleus is important in answering this question here, and I hope that made sense. I hope this video was helpful in understanding these topics. One last thing, I am a tutor. If you live in Southern California, feel free to contact me via email at moofuniversity@gmail.com. See the description below for more details. Thank you for watching.